Friends, today we are going to study major chromosomal abnormalities. There are different chromosomal abnormalities are there. So we'll know what are chromosomal abnormalities, what are the types of chromosomal abnormalities, what are the examples. So let's start with the introduction. Human chromosomes have a specific structure based on shape and size. You have already studied human chromosomes, which are classified based on the position of centromere. A chromosomal disorder results from a change in the number or structure of chromosomes. So about this is about the chromosomal disorder, which occurs because of change in the number of chromosomes or change in the structure of chromosomes. Normally, we know that we have 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes in our somatic cells. So this is the normal chromosome number in somatic cells of human body. But the chromosomes number may change. Instead of 46 chromosomes, there may be 45 or more, maybe 47. So change in the number of chromosomes leads to chromosomal disorder. Similarly, there may be the change in the structure of chromosomes. There are some structural abnormalities in the chromosomes which occurs because of addition of a segment of chromosome, deletion of the chromosome, translocation of chromosome, or inversion of the chromosomes. So we can classify chromosomal disorders into two types: numerical abnormalities and structural abnormalities. Half of all spontaneous abortions are due to chromosome abnormalities. What does it mean? That when there is a abortion is something what unable to progress into the pregnancy, right? So when there is abortion process, so that abortion, approximately half of all the spontaneous abortions are based on chromosome abnormalities. So if there is a chromosomal abnormalities in the fetus may not develop into a baby, and usually those fetus are aborted. The pregnancy is not progressed. Any deviation from the normal karyotype is known as chromosome abnormality. So normal karyotype, we have studied the karyotyping. If there is any deviation, means change from the normal karyotype, instead of 46 chromosomes, if we have 45 chromosomes, or if we have 47 chromosomes, or if we have missing any sex chromosomes, or if we have additional sex chromosomes, so we can consider them as a chromosomal abnormalities. While some chromosomal abnormalities are harmless, means when there is some of the chromosomal abnormalities, when it occurs, maybe because of number, maybe because of structure in the change in the structure, so it may be harmless, but some are associated with the clinical disorders, where we can see the signs and symptoms. We can see the changes in the body, changes in the shape, changes in the gender of the body, who suffer from the chromosomal abnormalities. Historically, scientists have used a staining technique that colors the chromosomes into a banding pattern. This is what we have learned in the karyotyping, where an approach of banding pattern or banding technique, we can color the chromosomes, and based on that we can be able to see if there is a change in the shape and size of the chromosomes. Also, we will be able to find out the homologous chromosomes, the partner chromosomes, or we can be able to trace out if there is a missing any particular chromosome number. These binding patterns make each of our individual chromosomes easier to identify, like a map. So this is what we see, that when we perform the banding technique, so banding patterns on the chromosomes, the light band and the dark band, helps you to differentiate the two different chromosomes, or also helps to group the similar chromosomes in a particular group. Here we can see numerical abnormalities, which is because of the change in the number of chromosomes. Here we see is Down syndrome. This is the syndrome, this is the abnormality, and this is the incidence, the prevalence. If you talk about Down syndrome, it is a trisomy of 21. What is a trisomy of 21? It means the 21 number chromosomes are not 2 in number, it is 3 in number. Usually the chromosomes are in pair, right? So instead of pair, the 21 number chromosomes are 3. Tri means 3, okay? So there is a 21 chromosome, okay? 21st number chromosome is 3 number. And this incidence of Down syndrome is, out of 10,000 population, 15 people suffer from Down syndrome. Similarly, Edward syndrome, the abnormalities is trisomy 18, which means the 18 number chromosomes are 3 in number. What is the incidence of this Edward syndrome? Usually, it's in out of 10,000 births, 3 are usually affected. Patau syndrome, where trisomy of 13 occur, and the incidence is 2 out of 10,000 birth. Turner syndrome, there is a monosomy X, means there is only one sex chromosome. Okay, usually the female has a 2X chromosome and the male has a 1X and 1Y chromosomes. But the person who suffer from Turner syndrome usually have only single X. So we say monosomy X, and usually the incidence is 2 in 10,000 incidence. Here the Turner syndrome, the phenotype is female, means the person looks like a female. Next is a Klinefelter syndrome, where abnormalities is because of double XY. So we can see there are 2 X chromosome and 1 Y chromosome, right? And here the phenotype is male. They look like a male, and the incidence is 10 people in 10,000 birth, 10,000 population. Triple X syndrome, where there are 3 X chromosome in the karyotype in the individual, and the phenotype of the person is female, and the incidence is 10 out of 10,000 birth. Here XYY syndrome, where XYY chromosomes are present in individual. The individual is usually male, has a phenotype, the gender is male, and this is the incidence rate, 10 in 10,000 population. Are you understanding?
Yes, sir.
Okay. So these are the numerical abnormalities. We can see that there is change in the number of chromosomes. There is trisomy of 21, it leads to Down syndrome. Trisomy of 18 leads to Edward syndrome. Trisomy of 13 leads to Patau syndrome. Monosomy of X leads to Turner. Double XY syndrome abnormality is Klinefelter syndrome. Triple X abnormality is because of triple X syndrome. XYY abnormality is because of XYY syndrome, right? Now here, what we are observing here, the most severe chromosomal disorder. This is the numerical abnormality is considered as the most severe chromosomal disorder. It is caused by the loss or gain of whole chromosomes. So there is either you can see, like example Down syndrome, there is a gain of one whole chromosomes. Because of that, the 21 number chromosome becomes 3 in number instead of 2 in number. Can affect hundreds or even thousands of genes. So here we can see that if there is a trisomy or monosomy, so in such condition it can affect the various genes present in that chromosomes. Usually fatal, so because it's a severe disorder, so it is usually fatal. Maybe it may lead to death. Oh, okay. This is what we can see. This is the normal male karyotype, and this is the karyotype of Edward syndrome. From the previous slide, you have seen that Edward syndrome is because of trisomy of 18. Can you see? The 18 number chromosome is 3 in number. Instead of, you can see all chromosomes are 2 in number, but chromosome 18 is 3 in number. So here we can plot a karyotype and we can identify what kind of disorder is there. Okay, we can compare normal male where one is, and you can see the sex chromosomes. There is a one X and one Y chromosome, and based on this, we say the karyotype is of male, and this is normal because there are 46 chromosomes are there, and all chromosomes are in pair, right? What we find in Edward syndrome, this is the phenotype is XY, so its genotype is XY, so phenotype will be male. And we find one extra 18th number chromosome, and so we call it as trisomy 18, and we the syndrome is the abnormalities is Edward syndrome. So karyotype image showing numerical chromosomal abnormalities. Now you can see Patau syndrome and Klinefelter syndrome. Can you point out the where is the abnormalities in the Patau syndrome? Yeah, one of you can say, can point out which
Sir,
chromosome?
13 chromosome में.
Yeah, 13 number chromosome is 3 in number. There is a trisomy of 13. So this is the Patau syndrome we call it. Okay. In the previous slide, we have seen that the trisomy of 18. So the trisomy of 18 is called Edward syndrome, and here trisomy of 13 we call Patau syndrome. Now the Klinefelter syndrome. Where is the defect?
Sir, last one में double
Last,
X.
yeah, there is one extra X chromosome. Okay, here we can see the genotype is double XY in the gender, and the phenotype is the male. Phenotype means the appearance, how it looks like from the outside, right? So by gender we can say the Klinefelter syndrome is a male person. So this is what again showing the karyotype of Patau syndrome and Klinefelter syndrome, which are numerical chromosomal disorders. Now let's know structural abnormalities. Till now we have studied numerical abnormalities. Here in the structural abnormalities we can see the change in the structure of the chromosomes. First we can see addition. Addition means when part of a chromosome is extra. So when we see a chromosome, so one of the segment is not normally present in that chromosomes. It might have added from the outside or other chromosomes. So that is a what addition means when part of a chromosome is extra, right? Deletion, one of the images has shown this deletion image. You can see like the definition is deletion means when part of a chromosome is missing. You can see in the deletion image here at the top left side, this is the chromosome which is present normally, and the part which is showing orange yellow color, so that part is missing, and we can see the size of the chromosome become very shorter when some segment of the chromosome is deleted, right? So usually it happens when there is a deletion, size becomes shorter. Now we can see duplication. Duplication means it's a mutation causing part of the chromosome to be repeated, resulting in extra genetic material. Right? So the left side, this is the normal chromosome, and we can see that the highlighted part in the chromosome is duplicated. twice in time. So when there is a duplication, the same sequence or same segment is duplicated. So now we can see the size of the chromosome become bigger when there is a duplication. And when there is a deletion, the size of the chromosome become shorter. Now let's know what is translocation. Translocation is when a part of a chromosome is transferred to another chromosome. So one chromosome part is transferred to another chromosome. That is called interchromosomal translocation. It's of two types: reciprocal translocation and Robertsonian translocation. What is reciprocal translocation? Let's know. Here we will see the segment from two different chromosomes are exchanged. Now on your right side, at the top right side, you can see the image is showing reciprocal translocation. There are two chromosomes colored differently. Now we can see there is this part of chromosome is transferred on the smaller chromosomes, and the smaller chromosome some part is faded on the larger one. So we can see there is a segment from two different chromosomes are exchanged. So exchange of the segment within the two different chromosomes. This is a reciprocal translocation. What is a Robertsonian translocation? Where an entire chromosome attaches to another. The entire chromosomes attaches to another. You can see here that two chromosomes are there, and these two chromosomes attaches to each other, and it has made a single chromosome. So this is called as a Robertsonian translocation. I hope you have understand the two types of translocation. If you have any confusion, please do ask so that we can discuss here. See the reciprocal translocation and Robertsonian translocation. The major difference: reciprocal translocation, where the segment from two different chromosomes are just they're exchanged. So this part of this chromosome is transferred to this smaller one, and the smaller part, a smaller chromosomal segment is transferred to the bigger one. So this is the exchange of this chromosomal segment. So this is called reciprocal translocation. Whereas Robertsonian translocation is something like, and when the entire chromosome attaches to each other and makes a single chromosomes. Here we will see inversion. It is also a structural abnormalities, which is a chromosomal rearrangement in which a segment of chromosome is reversed end to end. So this is the chromosome, and you can see the inversion process where the chromosome sequence is rearranged. Let's say here there is a B, C, D, E, and we can see we can write like there is a reverse inversion process. So E upper side will be E, then D, C, A like that way. यहाँ पे उल्टा होगा, ठीक है? अगर मान लीजिए यहाँ से A, B, C, D, E लिखते हैं, तो ऊपर ई वाला पार्ट इन्वर्ट होके ऊपर से आएगा, ऊपर से हम अब उल्टा काउंटिंग कर सकते हैं. जो इन्वर्जन होगा तो E, D, C, A ऐसे लिख सकते हैं. Okay, so this is the inversion where there is a chromosome sequence rearrangement, and with the segment of chromosome is reversed. So there is a reversion, reversed in the chromosome sequence. Now ring formation, when a portion of chromosome has broken off and formed a circle or ring. So here we see this part and the small part, the upper region has been deleted, and after that both the end will join and form a ring-like structure. So now we can see the chromosome which are usually linear. Now it's looking like a ring form, a circular form. So this is also chromosomal abnormalities. It's basically the structural abnormalities in the chromosomes. These are what the different chromosomal abnormalities based on the change in the structure. The first one is Wolff-Hirschhorn syndrome. Wolff-Hirschhorn syndrome is, what is a kind of abnormalities here? There is a deletion from tip of short arm of chromosome number 4. So here the basically disorder is in the chromosome number 4, what happened in chromosome number 4? There is a deletion from tip of short arm. So a deletion, there is a deletion of short arm. We have a two arm in the chromosome, P arm and Q arm. P arm is a long arm and Q arm is a short arm. Okay, or sometimes we say P arm is short arm, Q arm is a long arm. Okay, so if you here the P arm, the tip of the chromosome is deleted, basically in the chromosome number 4, and this kind of syndrome called Hirschhorn syndrome occurs in 50,000 births. So out of 50,000 births, one usually suffer. Next is a cry-do-chat syndrome, where there is a deletion from tip of short arm of chromosome number 5. So chromosome number 5, short arm, the tip of the short arm is deleted. Okay, and again the incidence is similar to the first one. Out of 50,000 births, one is affected. WAGR syndrome, where there is a microdeletion from short arm of chromosome number 11, and incidence is 1 in 5 lakh or to 1 in million. So out of 5 lakh to 1 million births, one is affected. Prader-Willi or Angelman, where there is a microdeletion from short arm of chromosome number 15, and the incidence is 1 in every 15,000 births. Dye-George syndrome, which is the microdeletion from long arm of chromosome number 22, and the incidence is 1 in 4,000. How do chromosome abnormality occurs? Chromosome abnormalities usually occur when there is an error in cell division, resulting in cells with too few or too many copies of chromosomes. So the chromosomal abnormality that we have studied, either it is a numerical or a structural. So this the reason of this structural or numerical chromosomal abnormalities. The reason is it lies at the time of there is errors which occurs at the time of cell division. When the cell divides, at that time, what happens? That kind of abnormalities that we have studied, the numerical as well as structural chromosomal abnormalities may occur. So cells may receive few number of chromosomes or may receive too many chromosome copies. Most chromosome abnormalities originate in the egg or the sperm. So here the chromosome abnormalities may occur in egg or sperm also. So egg and sperms are the cell which are haploid cell which has a half number of chromosomes. So each chromosomes are in a single form, not in pair form. So there may be the abnormalities before fertilization process, before fertilization of the egg with the sperm. So some happen. Sometimes the abnormalities also occur during the embryo development. Embryo is something like the when the fertilized egg is developed, is progress to develop, so it will develop into the embryo. So during embryo formation also the chromosomal abnormalities may take place. Okay, or sometimes these chromosomal abnormalities are inherited from the parents. Chromosomal abnormalities that originate in the gametes are present in every cell of the body. This is what we can observe, that if the chromosomal abnormalities occurs in the gamete cells like the egg and sperm, then that abnormalities can be present in every cells of the body. I hope you have understand what are chromosomal abnormalities. Okay. Now, any questions till now? Yeah, we can discuss the questions. Do you have any question, students? Are you there?
सर एक बार वो वाली स्लाइड निकालना जिसमें वो था टाइप ऑफ एबनॉर्मलिटी
Type of abnormalities. There are two types of chromosomal abnormalities. One is a numerical and other is a structural chromosomal abnormalities. This is the type. ठीक है? जो हमने शुरू में पढ़ा था, और इसी को हमने डिस्क्राइब किया न्यूमेरिकल में। हमने बहुत सारे एग्जांपल पढ़ा, डाउन
सर जो
सिंड्रोम।
स्ट्रक्चरल अबनॉर्मलिटी है उसमें जो क्रोमोसोम्स होंगे वो दोनों डिफरेंट डिफरेंट होंगे ना?
स्ट्रक्चरल में,
यस सर।
स्ट्रक्चरल में दोनों में डिफरेंस है कि डिलीशन, यहाँ पे देखिए डिलीशन वाला बात है,
सर इससे नेक्स्ट वाली स्लाइड में।
ये वाला
यस सर।
ट्रांसलोकेशन वाला।
यस सर।
ट्रांसलोकेशन इंटर क्रोमोसोमल होता है। इंटर मतलब दो क्रोमोसोम के बीच में होता है।
ओके सर।
दो क्रोमोसोम हो सकता है, होमोलोगस भी हो सकता है और हेटेरोलोगस भी हो सकता है। ठीक है।
ओके सर।
ओके, एनी अदर क्वेश्चन?